Good afternoon, everyone. My name is Csongor Váróczy, and I'm from the University of Debrecen. I feel really honored to stand on this stage, and at the same time, I'm truly delighted to see so many talented students in the audience. Now, in the next few minutes, I would like to give you a brief insight into my research activity and take you on a quick journey into the mysterious and fascinating world of cancer immunology. Breast cancer is an incredibly aggressive, malignant disease, and nowadays, unfortunately, 1 in 8 women need to face this scary diagnosis in their lives. But breast cancer is not only a mass of tumor cells, because if we take a closer look on a microscopic level, we can clearly see that cancer cells are always surrounded by a vast array of immune cells, creating a quite unique and complex atmosphere called a cancer microenvironment. And now I would like to introduce you to macrophages, which are one of the key players of the immune system and also the primary focus point of my research. Macrophages actually always remind me of one of the cutest and my most favorite childhood movies, Inside Out. You may probably know the movie where the thoughts and the actions of the main character, Riley, are controlled by these little colorful characters that represent her emotions. And this is what basically macrophages do, because they take part in the defense against pathogens, they also fight against cancer cells, and they aid tissue regeneration and various other healing processes in the body. Now, sailing to a little more scientific waters, macrophages can actually be divided into 2 major subgroups based on their immunological functions. The so-called M1, or classically activated macrophages, induce and maintain inflammation, they have aggressive phagocytic capabilities, and they produce large amounts of nitrogen monoxide and reactive oxygen species. On the other hand, M2, or alternatively activated macrophages, rather promote tissue regeneration and angiogenesis, which is the formation of new blood vessels. If a malignant cancer arises in the human body, a huge number of macrophages migrate from the spleen to the tumor stroma, and then they begin a crosstalk with cancer cells. As a result, they usually polarize towards the M2 phenotype, and they become so-called tumor-associated macrophages. This will then accelerate tumor expansion and metastasis formation, meaning a pretty bad prognosis for the patients. Although the exact signaling pathways behind macrophage polarization are really complex, a small signaling protein called interleukin-4 is known to have an essential role in the process. Our primary research aim was to study the role of the parp14 enzyme in the context of macrophage polarization. Parp14 is a member of the poly-ADP ribose polymerase enzyme family, and it has several regulatory functions, including the modulation of the interleukin-4 pathway. When the cell is in a dormant state, parp14 binds to promoters and prevents the expression of anti-inflammatory genes. But as the interleukin-4 pathway becomes activated, parp14 dissociates from the promoters and allows the binding of transcription factors, which will then lead to the expression of proteins like fibronectin, MRC1, and CCL17. We set out to develop various methods to influence macrophage polarization and the anti-tumor response via the parp14 enzyme. In our first experimental model, we differentiated macrophages and co-incubated them with breast cancer cells. The cells then spontaneously formed a 3-dimensional cluster called a spheroid, which closely resembles the cancer microenvironment. We treated the spheroids with a small protein called interferon gamma in order to activate the macrophages, and the next day we treated the spheroids with an enzyme inhibitor that is specific for the parp14 enzyme called MCD113. We measured the expression of M2 macrophage markers using RT-qPCR, and we also expanded our experiment with high-resolution confocal microscopy to monitor cancer cell death. These graphs show the relative mRNA expression of M2 macrophage markers. If macrophages exhibit the M2 phenotype, they begin to produce various proteins such as fibronectin, CCL17, and MRC1. If you take a look at the columns on the left side with the turquoise colors, they represent those spheroids that only consist of cancer cells and macrophages but haven't received any treatment. But those columns on the right side with the navy blue color represent those spheroids that were treated with the parp14 specific inhibitor. And as you can see, parp14 inhibition significantly reduced the expression of M2 markers. We also performed high-resolution confocal microscopy to detect and measure cancer cell death in the spheroid. In these fluorescent images, breast carcinoma cells appear as green, as they express the green fluorescent protein. Macrophages appear as blue, and the dead apoptotic cells are marked by a red annexin-5 staining. If you take a close look at the graph, you will see the column in the middle, which represents those spheroids that were only treated with interferon gamma, and the column on the right side with the red color represents those spheroids that were treated with the parp14 specific inhibitor. And as you can see, parp14 inhibition significantly increased the number of dead tumor cells in spheroids. We also wanted to gain a closer look of macrophage polarization in a human model. Therefore, we decided to test our inhibitor on primary human cells as well. We isolated primary human monocytes from blood products using a magnetic separation technique, and we differentiated the monocytes to macrophages. In this case, we pre-treated the macrophages with the parp14 specific enzyme inhibitor, MCD113, and we polarized them towards the M2 phenotype by adding a small protein called interleukin-4. We measured the expression of M2 markers using RT-qPCR. And as you can see, parp14 inhibition strongly decreased the expression of M2 macrophage markers, similarly to our spheroid model. In conclusion, our results have revealed that the parp14 enzyme has a dominant role in the M2 polarization of macrophages. Parp14 inhibition strongly represses the formation of the tumor-associated macrophage phenotype, and it also increased cancer cell death in three-dimensional spheroids. Taken together, these results suggest that parp14 inhibition may induce stronger inflammation and a more efficient anti-tumor response in the cancer microenvironment. Now, I would like to grab the opportunity to express my sincere gratitude to all of my mentors, including Professor László Virag and Attila Bácsi, Endre Kókai, and Isotta Sturniolo. I'm also incredibly grateful to Ákos Bede for his contribution as a co-author. He's also another really talented Szent Györgyi student, and he's now excitedly watching me in the audience. And last but not least, I would like to thank the National Academy of Scientist Education and the University of Debrecen for the professional and financial support. Thank you so much for your attention, and should you have any questions, please feel free to reach out to me in the coffee break.